Yes. Good morning. Good morning and very warm welcome to every distinguished guest from different organizations. We are at Agriculture and Forestry University for National Conference on Sustainable Agri-Food System, SAFS 2026, under the theme of Building Sustainable Agri-Food System for Food Security, Livelihood and Green Growth. This conference is co-organized by Ministry of Agriculture, Forest and Environment, FAO, for Care and World Hunger Health. The contributors of this conference are NARC, EcoHimal, Nepal Bank Limited, Kabi, Leeward, ADRA, Forest Action, AW International, Nabil Bank, Global IME and FAO Nepal. यहाँहरु सम्पूर्णलाई एग्रिकल्चर एन्ड फरेस्ट्री युनिभर्सिटीको नेसनल कन्फरेन्स सस्टेनेबल एग्री फुड सिस्टम SAFS 2026 मा हार्दिक स्वागत छ। आज हामी तेस्रो दिनमा छौं, तेस्रो अथवा अन्तिम दिनमा छौं र आज हामीहरुको केही स्केड्युल अथवा हामीहरु सबैसँग छ भने आज हामीहरु को ओभरअल स्केड्युललाई म थोरै छोटकरीमा भन्न चाहन्छु। आज हामीहरु 6 वटा किनोट प्रेजेन्टेसन गर्नेछौँ। हिजोको एउटा किनोट प्रेजेन्टेसन बाँकी छ, त्यो सँगै 6 वटा किनोट प्रेजेन्टेसन छ। त्यसपछि हामीहरुको ओरल प्रेजेन्टेसन्स प्यारलल सेसनमा जसरी हामीले हिजो र अस्ति गरेका थियौं र त्यसपछि हामीहरुको पोस्टर्स प्रेजेन्टेसन. So we have the oral presentation parallelly in the 3 respective halls like yesterday and the day before yesterday, and we are going for the posters presentations. Overall poster evaluation हामीहरु सबै छ, त्यो पोस्टर इभ्यालुएसनको कुरा पनि छ, त्यो कुराहरुलाई पनि लिनेछौँ भने त्यसपछि हामीहरु क्लोजिङ सेरेमनीमा जानेछौँ आजको यस र त्यसरी हामीले यो 3 दिने नेशनल कन्फरेन्सलाई कन्क्लुड गर्नेछौँ. सो म सम्पूर्ण डिस्टिङ्ग्विस्ड गेस्टहरु र यहाँ आउनुहुने सम्पूर्ण अतिथिहरुलाई हार्दिक स्वागत गर्दै यथास्थानमा आफ्नो आसन ग्रहण गर्दिनु हुन र अब किनोट प्रेजेन्टेसन्समा कन्सन्ट्रेट भइदिनु हुन हार्दिक अनुरोध गर्न चाहन्छु. So, for the first presentation of today's keynote presentation, actually he was scheduled for yesterday's presentation, but he couldn't make it to the time because of transportations and other factors. So, I would like to request Professor Dr. Zheng Thie Yang, give a round of applause, from the National Chung Hin Sing University. He is the activist, emeritus professor. He is the entomologist with expertise in insect biodiversity, taxonomy, agricultural research, academic teaching, and international scientific collaboration. As we already mentioned that we have only 15 minutes time for the presentations, I hope Dr. Zheng Thie Yang will manage it within the limited time, within the given time. So the stage is yours, Professor.
Yeah. Okay. Thank you for the organizer, the invitation and the introduction. Because of the time is for me, I'm sorry I missed the regular ordinary schedule because of my flight planning, something program. And this morning, I just used very quickly to introduce something like just one idea to make everybody knows sustainable, is maybe not so sustainable itself. Because I remember in IUCN meeting in Jordan, Amman, we are SSC, sustainable species group meeting, and our chair just asked if we are sustainable, how about our subject will be sustainable? But I'm happy now, still sustainable, and merge to every aspect, include our food, our agriculture. Of course, at that point, that's because of the environmental sustainable. But now, our human, we are concerned about if we are sustainable or not. But today, I will also jump one step earlier more than what we are talking about the topic of the conference. It's not only for the product, for our life, but if you want to do something more better, you think about, when we're talking about the IPM, we have to do the identification more definitely, and you can make sure you are doing the right species. We have a lot of species, a lot of the case, happened whenever you invest large money or maybe the manpower and the time for 10 years and maybe 50 years, then you find what you are studying is the wrong species. That means, you waste the money and wasted everything. But the other thing is because of the trade-off, trading and transportation now getting, developing and developed. So the agricultural product transport from here to there, not only the inland, but internationally. So the insect pest, must be, propagate not only by the natural way, but also by the human, the help. So in this way, I would like to give the case very briefly. The apple worm is very important quarantine insect pest to Taiwan, and also to some area that they don't have the plantation, only they eat apple. They don't want to eat the apple with the worm, the coating moss, right? So in this way, actually I just want to say, if taxonomy, is important, that's because of we have to make this, the protocol for identification, not only by, maybe everybody knows there is one app called the color and the form, and you just guess. Guess this is the species, but maybe it is wrong, right? So if most of the identification or the resources survey, we start from the species by species and the name by name, and finally it becomes a biota or fauna, flora, and the others like micro fauna, whatever, right? So this is sorry, it's in Chinese, but never mind. You have a lot of different resources for you to do such kind of the process. And the point is the process from the bottom is a dissertation thesis. Most of you has one copy at home, maybe never published, right? And then you have to have some taxonomist to find out your thesis and published as the paper. Well, the first you need is the checklist. Then the new text happen. You published. Then after some 30 years, the name list may be changed. So you still use the old name, like here I know some apiculture, Apis mellifera, yes, Apis mellifera linguista, maybe nobody knows, but this is the subspecies. But several subspecies of the mellifera belongs to the genus Apis, but always change a lot because now we have the molecular, and someone maybe find the AI to review for you. So in this way, we have to go more upper. It's some 100 years, you have the new monograph to publish, new book. And then you publish it again, the new. But nobody knows the new name because since 1753, Linus published the first plant species. Then 1758, then published the first new animal species. Then, until now, so many things happened. It's a million, right? And then, if you have such kind of a publication, then you have to think about, easier for the layman, the common people to understand. So the illustration and the description is the last. But now everybody can find from the internet, the illustration, the picture, and the figure. Then you look at the picture and the figure. You say, okay, this is which species? Actually, this is only guess, right? Like you see so many green, you say this is plant, right? You see the red at the beginning, you don't know this is a flower, so you don't know this is also plant. You think this is something else. So in Linus, the cockroach, the larva, and the adult, looks similar, so they call it the same. But to the caterpillar and the butterfly, they may be identified as different species. Okay, in this saying is actually what I want to talk to you is, the case just finished because you just guess this is apple worm. But so far, maybe nobody rear the insect life cycle, then identify all different development stage. So I just show you what I was doing, right? Okay, this is mostly biota. This is a fauna of the hexapoda in India, right? Just for case. And before IPM, the first step is quarantine, because if you can prevent from the insect to go into your territory, that's your success for the first step. So the quarantine important things is the lowest part, right? So must be training, and practice too, right? So to prevent the insect come and the identification. So we identified the apple worm and prevent the apple import from the USA. Then we get a chance to import from Chile, from Australia, from other countries, right? And this is because of the WTO, right? Okay, and this is a case I want to tell you. Whenever identification is just something like this way, but actually, what you see is a mature larva. If it is not mature, you cannot identify, okay? And you cut a lot of the apple, the right upper, you see? And maybe no worm inside, but it was already, infected. So you cannot identify the insect injury, then say this is a species, okay? And finally, we have to see the detail. Okay, just go through. Like the left upper, it's already get into the pre-pupa. That means they are going to pupation, but you cannot identify this one. So the timing also very important, right? And the others, not the mature, is also not able to, right? And again, those damaged part, damaged symptom, but you cannot identify this without the identity, because the taxonomy is an identity sign, right? Okay, so you have to do a lot of work, and all the material you sampling from the cargo import to your country, so you can identify. But actually, I have to do evidence, review from the field. So I go to this is Hamburg University professor there, and just in his backyard, we collected the apple like this way, right? And we do the same thing. And finally, we find, this is, you identified this is the insect, yes. The worm, the codemouse, yes. But the name, and you find from the internet, but you cannot find the morphological evidence, right? So we have to do the different thing because all the molecular, the identification, right? So this is we find the way we find the character to identify, and we finally find that actually we think they are the same species, just only 11 specimens, but actually they belongs to, two species, right? So we were wrong. Maybe, contamination. Maybe both are quarantine important, right? So in this way, mostly the symptom you check from the stock or the novel, right? And again, so we know the character is actually on the leg, pro leg, right? And finally, we find it totally like a 10 character. But when identification necessary, they say, okay, if you can identify 5 character, that means 50% correct. 50% correct, that means you said yes or no, right? But this is we find the first time we published all the character. But actually, some character are still not very clear. Okay, we go very quickly. This is the adult specimen for identification at the beginning. Identify to the species by this way, but they never had one to publish in the larva the stage to publish the species. So so many species. Which one is the codemouse, right? Because this is illustration. So they are not naturally find from the field. So you also cannot find from the field what it look like, right? So this one is the one, right? And I just go quickly to show you, So previous study focused on the adult morphology and character, but no one to do the larva. The insect larva is something like this way, right? Some different odors, very, very different. And this is what we just show. The pro leg is the important one. But this time include the anal leg we also used. And then, okay, all the character, what we just want to use is the crochet, the leg. The larva can attach the leaf because they secretion the silk on the leaf surface. Then their leg with the claw can climb in, on the slippery leaf of the plant, right? Okay, so this is the way, the publication from the textbook since 1960 some until now, the same picture, never have the new one, right? Okay, so this is all terminology. I don't want to show you, but in the workshop, some days later, then I will introduce in the department entomology. Okay, so this is we took the picture from the codemouse. They are circular. The left one, right, is the right one. That's different family larva. Okay, and we still have to check this hair. You even cannot see, but we have to identify. This one. So if you cannot see this one, that event, you cannot identify and tell the name. But actually, you have only one insect specimen for your identification, because they checked from the cargo by cargo, a lot of the sample, but only they find the one larva and not the pupation and not the immature, right? So in this way, you still find the spherical and the position, the morphology, right? Okay, I just go through for you to see how detailed compared to the molecular DNA, right? But the DNA, you cannot see anything. But this one, you must check out. Even I myself use the hand handle microscope, right? The very, very magnified. You have to check. Because this is the way you collect the basic data, right? So I publish the character, then you check one by one. And whenever you get an 8 character meet, then you say this is 80% correct, right? So this is, you have to check. That's a different species. And the larva, they say the color, but actually their values are diversified, right? And the head, the marks, they will show you someone, just the identity, but not the variation. So you have to show the variation by some way. Every leg you check. And finally, we use the very basic, covariance to see the sample is unified or not, right? Then we find the second leg, the right side, and the third leg, and the right side is very trustable, right? Okay, so then we find the normal distribution and find actually the other species, right? The white one and the black one, they are different species. You merge together and find your identification was wrong. Maybe not wrong, but not trustful. So if you are not identified by the taxonomic way, taxonomic protocol, then maybe you have to check back the species what you are working on. Maybe not the correct species. And this is what I want to say. If everybody think about very, very, very frontier working, and you have to think about, they are raised from the basic, from very beginning. Since 1758, the first animal species published, you think about the name change a lot. And actually, that's because of you get the wrong idea. You use the modern science to study the very traditional subject. Thank you all. Thank you. Sorry? Any question? Our chair is coming.
Thank you, Dr. Jain. So we'll take questions and discussions later on of the lunch time.
Okay.
Because we are running out of time. Thank you. Thank you, Professor.
Okay.
So now, moving forward, I'd like to request Professor Dr. Madhav Pandey, Department of Genetics and Plant Breeding, Faculty of Agriculture and Forestry University. He is a pioneer wheat breeder with extensive experience in wheat research, teaching, variety development, crop productivity enhancement, and food security. So I'll request Professor Madhav Pandey to be on the stage and 15-minute time for his presentation. Thank you.
About the slides. Okay. Thank you. A very good morning. and namaste to all the participants of this session. The title of my talk today is A Journey of Wheat Breeding Research at Agriculture and Forestry University, focusing on achievements and future perspective. My presentation will be more of storytelling and less of science, so please forgive me, particularly those from the foreign participants who are very much familiar with breeding research in the academia, but the scenario here in Nepal is a little bit different. We are not that much involved in breeding research in the academia, so for the domestic in-country participant it can be interesting, but for the foreign or the from other nations, including our neighbors, it can be boring, so please forgive me for this. Thank you. First of all, I would like to give a little bit context about why we started plant breeding research focusing on wheat improvement at Agriculture and Forestry University. As we all know that the reputation of agriculture universities around the world are mostly judged not only by the academic programs, but also from the various technologies developed by them, like crop varieties, production technologies, and etc. The university breeding program not only develops the new varieties as a direct output, but also excellent opportunity for hands-on training and genetic materials for student training. So the long-term wheat research was initiated at Agriculture and Forestry University in 2016 with the objectives of developing next generation. I'd like to emphasize on next generation a little bit, crop varieties for high yield, climate resilience, and better end use. So the idea emerged from then registrar at that time, Professor Surikant Ghimire, and research director, Professor Navraj Devkota sir. Both are my seniors and were teacher. And the directs, they invited call for long-term research on various disciplines. And in discussion with them, I decided to initiate wheat improvement research as a long-term research work. The mission statement that time we put on was to improve the life quality of Nepalese farmers, enhance farm income, and food availability through the increased wheat productivity, especially in the stressful production environment in the context of climate adversities, by the adoption of high-yielding, climate-resilient disease and pest-resistant varieties that are developed using the latest technologies. Because in our national wheat research program, mostly we rely on conventional methods, and keeping pace with the changing environment, production dynamics, we thought we must adopt new technologies in wheat improvement. A little bit of scenario in wheat research and breeding. Actually, the National Wheat Improvement Program has been highly successful and rewarding in Nepal, letting almost over 96% in-country wheat demand met by domestic production and the use of domestic varieties, which is actually worth praising. And the growing wheat demand and in-use quality requirement by milling industries nowadays in the present context are creating wheat improvement opportunities, and the future challenges lies to stagnant in yields because of different factors, including the lack of high yield potential varieties and disease risk, particularly rust, and climate adversities, frequent drought and heat stress. So this is the variety outflow trend in Nepal. So you can see it is not that smooth. So sometimes peaks and sometimes it go down and again. So the idea of showing this slide to give you an impression that despite the highly rewarding wheat improvement research in Nepal, the consistency and the consistency as well as the regularity of or the continuity of the research work, they can see some question mark we can see there. So that is why our university work can be supplementary or in some cases even complementary to the national wheat research program. Wheat improvement research portfolios, when we started the long-term wheat research at AFU, we had three portfolios. The first is the development of high yield potential, which is 7 to 8 ton. I mean that you can compare with our neighbors like India or China. China is actually far ahead of even the whole region, a little bit behind to Europe, I guess. But our varieties are, in some exceptional cases, some varieties at some research location may have 7 or 8 ton per hectare yield, but as a, in an average, yield potential is not that high. So when the yield potential of variety is low, then at the farm level that even goes down. So if we can improve the yield potential to a higher standard, then the farmers, at the farmers level, the wheat productivity can be enhanced without any additional inputs. And the second portfolio was for the breeding stress resilient, focusing on heat combined with drought, with yielding ability superior or at par with the commercial varieties. And the third portfolio was the germplasm enrichment and pre-breeding, some pre-breeding activities. So these were the strategies. Firstly, we relied on the international nurseries, advanced line and germplasm as a fast route. And then we also did some hybridization and developed local materials with some smart crosses and selection in large segregating population. And integration of modern tools and approaches like selection for gene QTLs, physiological trait, root traits, and DNA marker assisted breeding. So we were successful in getting collaboration from different renowned organizations like Chengdu Institute of Biology under the CAS, China, NARC obviously were our national partner as well as collaborator, CGIR, Excellence in Breeding, which we got some help for marker work and high density genotyping, CIMIT for Mexico for material, and also from the Washington State University we got some genetic materials, and Professor K.S. Gill, he visited our program at our university, and we have some very useful DS lines from his program. So this is the overview of wheat improvement research. We started in 2016, and all the ways this is exactly the folders in my computer. So if for each year we have the experiment, and we are up to 2025, and we completed one full cycle, and we are entering into the second cycle. So the first challenge was to develop a network of field test sites, because being a university, unlike NARC, we don't have satellite stations, research centers who can collaborate with us for multi-location trials, which is pivotal in the development of plant varieties for assessing the stability and the performance across the target environment. So we have from here, province, Madhya province, all the way up to Lahan, and then coming up to here in the far west, Dhangari side, across the Tarai region, we were able to develop a network which included our constituent colleges, some rented farmers' fields, some private companies, research fields, and even some special farmers' field where we trained people for data recording, and we visited frequently the fields and can generate some data that can be used for varietal release purpose. So this is the view of the fields. You can see, especially this is Professor Hanchinal from India on the far right slide. He was very much happy to seeing at our field, and really wanted to see all the material and come, and he says, okay, thank you, I'm going, and then a little bit goes further and then comes back again and you know, feels the varieties, and it was very much encouraging for us at that time. And here you can see we have different friends from collaborators from NARC, Lakshmanji and Dr. Hirakaji. So fairly, this is the picture of a field where we rented in the farmers, rented farmers field, and we put fence and planted some border crop, and were able to conduct 3 years multi-location trials there. Again, multiple field experiment sites. On the far right, you can see a Lumini Seed Company site at Bhairava. This one is IES Khairani campus. We also had a trial there. And to highlight some of the achievement while completing the first cycle of long-term research and wheat improvement, we have identified 2 candidate varieties which will be very soon applied for release or registration, depending on our priority and the situation. And pre-release deployment of farmers, deployment for farmers response study of candidate varieties accomplished in 2024 and 25. In the last 2 years, we extensively distributed the seeds of our candidate variety as an experimental material and grown in an extensive way to see the farmers' responses as well as the varietal performance. And we also did seed multiplication candidate varieties in collaboration with private seed company, Lumini Seed Company in Bhairava, which has been very instrumental in multiplying the seeds, which is one of the major activities in any varietal development program. And also we developed around 8,000 advanced lines, which were bred or developed here at AFU with our own effort and our material, which are targeted for high yield, drought, stress, tolerance, rust resistance, and for a better end-use quality. And we also have developed a germplasm pool of around 200 to 250 lines that comprises our material, advanced lines, some material from China, DS line from the USA Care Skills program, so which will be very much useful in the future for our wheat improvement program. And also germplasm enrichment, which also includes the recombinant inbred lines and DS lines, as I mentioned before. And with this, another salient feature of this wheat improvement program at AFU is the thesis research of over 30 MSc students from plant breeding, biotechnology, agronomy, and plant pathology department, and also a few bachelor's students in the LEE, which is a kind of internship program. So they have also worked on our material in the field, and they got a practical hands-on training and experience, a lot of exposure in wheat breeding. So this is one of the candidate variety AFU 001. The technical identity is MIT24, field at Rampur. And this one is another one, AFU 002, MIT08, again field at Rampur. And the UPS of candidate varieties, what makes different these varieties, I can mention that the AFU 001 is really highly uniform, and this is not technically, I mean, anything looking beautiful matters, but sometimes, you know, the farmers are very much attracted to the crop, and it is obviously, if not a big plus, but it's additional, you know, feature. And have high yield in favorable hard type, excellent in-use quality for millers, which we have test results. And harvest slow roasting genes, LR46, LR67, LR68, and LR37 complex. The all gene complex are slow roasting and pleiotropic genes, which not only works on leaf rust resistance, also for striped rust and powdery mildew. And another candidate variety, AFU 002, is high yield under late sown heat stress condition and longer grain filling duration. This has a really a salient feature of this variety, which distinguishes the variety from others, and it has been, you know, proven in the farmers' field as well as in our field. So it again harvests the almost same type of rust resistant genes like AFU 001, and it has some level of drought tolerance and hard type, excellent in-use quality. And this variety, AFU 002, was planted in about 6 bighas of land in our agronomy farm, west from the mid north-south road. If I mean the students and the participants from AFU can imagine or they visualize the location, and that is the area is devoid of any irrigation, and we planted in almost January, first week of January, and with no irrigation, nothing, just average fertilizer. And this year was a bad year for wheat production. No rain during entire, you know, until the grain filling, and then there was a rain which partially damaged the crop. Still, you know, the farm was able to harvest about 60 tons, 60 quintals. Yes. So that was not really high yield. We assume that it's around 2.5 or something. But compared to other variety in the farm like BJ, that did really well. So this is another proof for the UPS of our variety. And we also developed a package of practice info leaflets while distributing seeds to the farmers. And the pre-release deployment on-farm yield assessment, we did some on-farm yield assessment in the farmer's field. This is in the East Chittagong, and you can see the yield advantage here. It is under the normal sowing condition, and another field crop cut also showed yield advantage of AF000T, and with this location there was excess moisture during the planting, which is becoming a major problem in some of the isolated areas where people plant rice, and if the rice is late or there is a late or rainfall during October, or then there is a field is wet and it's not ready for wheat planting, and the farmers, they have to fit with the planting window. So they plant, but because of excess moisture, there is a germination problem, and the wheat crop is, that it is not for adapting the excess moisture. So that is bad. And they, while we were doing this, the farmers, they use one Shriram variety from India, unofficially brought variety, which they say is tolerant to excess moisture during the planting. So we hope this variety can give some benefit to our farmers. So we also carried out some interaction programs with the farmers. This was the right one from this year. So it was like a farmers training as well as interaction before distributing the seeds. We briefed them about our varieties and production package of practices, and farmers were very happy, and they liked, these kind of programs, and they were, thankful to our university as well. And again, the pre-release deployment, this farmer is from Far West, in the Tikapur area, was very happy. They have actually started keeping of the seeds for next season. So even before release, which is, for personal use, it is not a problem. Now coming to the latest technology, we try to integrate genomic tools in wheat breeding at AFU. Some of the tools we used while breeding the candidate varieties, I was just before talking, to identify the rust genes and some of the drought resistant and other important genes so far available, the markers available. And we also developed the varietal fingerprint for release purpose and gene tracking, and a CASP, STS, SNPs, indel for gene QTL identification, and mid-density genotyping for genotype fingerprint, as well as gene profile studies in varieties and advanced lines. For this work, we got support from excellent CGIR Excellent in breeding program. We did 2 times in 2021, I guess, and 23. So we have a good database of the genes and the QTLs and other random marker profiles of our Nepalese wheat varieties, our candidate varieties, as well as the advanced line. So this is the picture you can see. This is from our own lab. So here you can see the candidate varieties have different allele profile that makes our variety different from the majority of the varieties from National Wheat Breeding Program. And if you see again the phylogenetics, this is with the mid-density genotyping, 3,000 almost genes, markers actually, SNP markers, and you can see the BJ, Gautam, and this is Jingong 1, and this is our candidates, Jingong 2 and Bola. So this is, and with the trait-based marker, 400 almost trait-based marker, here the two candidates are in a different place, 24, and I guess 8 is here, where in the random marker they are coming together. So this shows the genomic diversity of our candidate varieties, as well as they are different from the majority of the wheat varieties in Nepal. It is important because while we are talking about the resilience, the biotic and abiotic stresses, the genomic diversity is very much important. That does not directly assures that your materials are, they will perform better or so on, but they will definitely give a basis for further, improvement and creating the diversity of working gene pool. So that is again important one. So here the target gene for marker estate selection we have identified is LR34, LR46 complex, LR67, LR68, and this one is a DRB1. This is for drought, and this also for drought. So this is under study, and CRE, this is nematode resistance. These two we are not ready to use, but they optimize marker system we are now planning to use for marker estate breeding. So again, this is for LR34. Here this one is the diagnostic, the BJ positive this did. And for LR46, the PAW1 is the positive control, and MIT26 has LR46. So the marker estate gene pyramiding, we have done something, a basic preparation for marker estate gene pyramiding for LR34, LR46, and LR68. All are slow roasting adult plant resistant gene, and if we can, this gene in single may not be practically useful, but if we combine it with other slow roasting gene or some other minor genes or QTL, then they can impart near immune or a practically useful resistance to rust. So we have F3 plants and progeny rows carrying 2 more recombinant genes were selected, and marker estate selection F4, F5 progenies. We have identified plants carrying this multiple desirable genes. This was actually 2 years back, so we have advanced our material. So our materials are already F6, F7, almost stable condition. So now we have to screen this material for the genes, recombinant genes, and identify lines that possess multiple resistance genes of this complex. So in summary, in the history of 60 years agricultural education in Nepal, the crop breeding research within the academia demonstrate competency and future promises. Nepalese farmer can directly benefit from these varieties. Scientific knowledge and the expertise, genetic materials are highly relevant to National Wheat Breeding Program for the development of stress-resilient, better induced quality wheat varieties, and breeding research at AFU was instrumental for student hand-on skill training and postgraduate thesis research, which I already talked about. So now it is, we go for a little bit challenges and future perspective. There are big questions now that shall AFU continue wheat breeding research and or initiate breeding research on other crops too? For example, hybrid maize, rice, barley, minor crops, vegetables. Because we have, farmers and the industry people coming for collaboration and looking for breeders who can work on these topics, can be interesting. Are the available infrastructure and expertise at AFU sufficient? Definitely not. We need to work more, particularly the farm and seed system, seed supply system, seed multiplication system is very weak. And for that matter, we have to work more. And currently, approximately 10 to 12% research projects at AFU are plant breeding related, 10 to 12. I want to mark that. And does the breeding research pays off? Obviously, I would say yes. So this is, if we compare the universities, other universities from India or the comparable universities, this is University of Agricultural Science, Dharwad, which was established in 2010, almost same year, like that of AFU. And actually, our Vice Chancellor, Registrar, that time they have traveled to this university and see their activities, and it was one of the purpose was to compare AFU with them as a benchmark. So if you see here, almost 42% research focus area, thrust area are devoted to plant breeding research. And again, another university in India established in 2010, Bihar Krishi Vishwavidyalaya, almost 34%. So this simply gives us that we are lagging behind. So we have to devote more research resources and effort in breeding research. That can give a university a output and a measurable impact on the society, apart from regular academic activities. So future perspective, briefly, I'm not taking too long time. Breeding research, you can see the changed climate, the climate model people, they always show this kind of things. So different colors means it is changing, yeah? The conclusion is it is changing, heat, drought, rainfall patterns, wheat import, in 2025, 90,000 tons, and was a chaos actually. And this is again one important thing is the risk of rust diseases. Why? So this, the slide from NWRP Bhairava shows that usually the leaf rust is a problem in the plain area, not in the hill or mountains. And stripe rust is the problem in the hill and high mountain area, not in the plain area, in the hot areas. But because of climate change or whatever you can say, there is a trend that the leaf rust is moving upward and the stripe rust is coming down. So nothing is now geographically, a zone there you can forget the disease, either of the disease. So we need to work more. And another important thing is the Barbary wheat rust paradox in the Himalayas, Nepal. This guy is a very renowned guy, Eugene. He works at University of Minnesota in the US. He's an excellent professor. He actually visited us in this summer. And we participated in the rust survey. This is the Barbary, and Nepal is a hot spot for Barbary diversity. And diversity is the alternate host for striped rust, stem rust. So it is not only for our country, it is for the global region. We have to work more on rust and Barbary connection, as well as the resistance. So there is always a possibility for work more. And we have new initiative. We just admitted two young PhD students. He's a wheat breeder at NARC. So he will work on genetic dissection of rust resistance in Nepalese wheat breeding panel and marker-assisted pyramiding of resistance gene into elite background. This project is supported by Food Security Education Project. So it, about 40 lakhs. So the resource is reserved. And for this project is for the excess soil moisture stress is a novel work. We are looking for funding for this. So again, acknowledgement. Here I would like to emphasize the Food Security Education Project for their support in the PhD projects. And before thanking you, can show you how we work and in the field, how we enjoy working with the farmers and the in the rural area. And thank you very much for your attention.
Thank you, Professor Madhav sir. Though it is a little bit lengthy, but I think it is worthy for showcasing what he is doing in Agriculture and Forestry University.
Okay. Hello? Hello? Okay. Mic कतातिर यता तलको Good morning, one and all present here. It is always good to listen to other presentations, actually. I enjoyed two presentations that was presented today. And first of all, I would like to thank the organizer for inviting me. And since yesterday I knew that the time is so limited, so I focused myself. In the title, I have given the presentation, covering all fisheries and livestock sector. But I'm now focusing only on the genetic improvement of animals for sustainable agri-food system. Enable गर्न even enable गर्नुहोस् please. Hang भयो जस्तो छ
यो रासन नलागेछ
Katya. Okay. Huh? Yeah, it is. Hello. के हुन्छ साउन्ड पनि आयो So it is taking time because of technical problem here.
What's the problem? Maybe you just speak, don't use the A. Right? Right? Just say something.
Okay.
सिक्युरिटी वार्निङ हुने
ये क्लोज गर्नु भयो
हेर्नु म त्यहाँ तल
Control P गर्नु भइसक्यो भएन भयो त skip
Okay. Yeah, sorry for that. Again, welcome you all in my presentation. So, as I mentioned, I'm just focusing in the genetic improvement of animals for sustainable, for sustainable agri-food system. So I'm just focusing on that, and my presentation will be more on the sustainable agricultural system, genetic improvement of animals, importance of genetic improvement for SAFS, and also conservation of animal genetic resources in the aspect of SAF, and what are the challenges and opportunities that we will have, and how do we go together as a collaboration. So when you think about agriculture, so agriculture plays a crucial role in the economic growth, accounting about 4% in the developed country. But if you see the least developing countries, it contributes around 25%. Yeah, that is also applicable for the Nepalese context, because agriculture contributes that much. So it also contributes more in this food security and livelihood. So when you see the agricultural development, it's a very powerful tool to end extreme poverty and increase prosperity and feed projected 10 billion people that we estimated in 2050. So during that time, and with the science, with the development of science, we assume that the longevity of the people are even, getting better. So in 2015, there will be more people who will be more than 65 A's. More than 65 A's will be like 1.5 billion people, which demands for the protein, high quality protein, that is especially from the livestock sector, that is contributing from the livestock sector. So when you see the regional, when you see the region, it will be the developing countries who are like contributing for this demand again, because for the developed countries they are already having the sufficient amount of protein they needed, but it's the developing countries which we need the demand, and we need to improve the livestock sector for attributing this demand of the demand of the livestock proteins. So when you see this thing, I'm just showing these slides to show that how it is, the scope of the livestock sector is increasing, because you can see the demand in every aspect, like in buffalo meat, in sheep, goats, in pig and poultry, everywhere this is increasing. So and again, when you see the reason, it's the poor continents, maybe like Asia, Africa, that's the one who has like the increasing scope for this aspect. So we are always thinking of how to fulfill the demand. So we are thinking of sustainable agriculture food system, always which ensures the food for the produced efficiently by protecting the environment, by conserving the natural resources, by supporting economic viability, promoting social being, and meeting present and future food needs. That means we are going to the environment sustainability, economic sustainability, and also the social sustainability, like cultural. If the culture demands something, then there should be cultural sustainability for sustainable agriculture food system to retain. So when you think about the role of livestock for sustainable agriculture, it contributes through food production. As I mentioned, it's also contributes in the nutritional security, food security, nutritional security, economic for the livelihood of the people, and also for the organic manure. And there's a risk management for rural households. So it is the livestock sector where more than 1 billion people are involved in, and also significantly contributed to the agriculture GDP. That is around 24% in our case. So, as I mentioned, like we have to have sustainability in different aspects. So, for that, genetic improvement plays a vital role. So, it is genetic improvement. What is genetic improvement? It is the systematic selection and breeding of animals with desirable genetic traits to produce offsprings which enhance performance, productivity, healthier animals, more efficient reproduction, and adaptability to the specific areas. So that improves milk, meat, and other traits as well. So for getting this genetic improvement in the population, we need to have, use different tools such as selection, which is a very sustainable tool which improve the sustainable development, improvement in different traits of the animal. And crossbreeding, performance recording, we need to have very systematic performance recording, and for that, we also need to have different modern assisted reproduction tools, and also we have to follow the new technology, new advanced technology, which we, in addition to the performance recording, we also have to have genomic selection together to have the more accurate and more accurate sort of decision making in the population. So I just want to tell what NARC is contributing for the sustainable agriculture, agri-food system. Actually, NARC, in the terms of animal breeding, genetics, and reproduction, we have been working in doing the genetic selection, genetic improvement to produce the high genetic merit animals. And we are working more in the indigenous animal genetic resources, which has very much positive attributes, such as adaptation. The adaptation trait is very important, which affects the production of the animals. And also we are doing some research on developing the breed, with the traits that we are focusing on, and also not only the breed that helps in improving the production, we also need to work in the animal health aspect, animal nutrition, and other management aspect. So we are also working on that aspect to improve the products and productivity and production of the population. So again, in order to, whichever the technology we are developing, we need that to be having the impact in the targeted group. So we also working on the socio-economic aspect and assist the policymakers to include that technology in the policy strategy. So when we are talking about genetic improvement in Nepal, we have taken the genetic improvement strategies, such as we do the importation of germplasm. We bring the high genetic merit bulls, which is of pure breed, and we bring the high genetic merit of cows, of the pure breeds again. And when we bring the semen, we bring the semen of the proven sire. Proven sire means their daughters are also performing very well. That is already proven. So we import the proven sire and also the tested sire. The tested sire are those which mother and father are the good performers. So mother and they are good performers. So those animals are also, the semen of those animals are also brought in to improve the population of the livestock. And also, these days, we are also bringing the embryos of the pure breeds so that we can have the, we can have the good herd of nucleus herd of a certain specific breed, so that we can do the genetic improvement in a faster rate. So. Not only that, these days we are also working on we are also having the genetic improvement programs where we can have we can select the high outstanding animals which has having the high genetic, high estimated breeding values, and we are using those the males of those for the semen production and using those. So NARC has initiated the very system and initiated and established the systematic pedigree and performance recording system in Nepal since 2008. Earlier, we don't have that kind of systematic system, but from 2008 we have been working on that, and it's a very successful project that was conducted in genetic improvement in cattle. So in this presentation, I will be giving some examples what we have done. Not all the activities, all the programs, all the projects that we have been working, but we are giving the examples for each thing. So this is the very important example that is done, initiated by NARC, and with the collaboration with the department, we worked together, and it became a very successful project. And it was done in 14 different districts, where we selected best animals, best outstanding females, and used the proven sire to have the future bulls for AI and for artificial insemination and natural services. So this is what we have done. We have used the open nucleus scheme, nucleus herd scheme for this one, and within the short period of time, not only we use the high genetic merit, we also use the different technology, intervene different technologies of management in terms of health and nutrition. So within the short period of time, we doubled the production that was in the beginning. Now we have different projects that is continuing in this aspect, like NLSIP, Milky Way project that is in Sinduli. There is Adra Torai projects, sapling projects, Jersey Valleys Nepal project. These all projects have follow the same DCIP, that is very famous DCIP projects strategy. But in this one, we have improvised the digitalization in the livestock farming. So with the digitalization, we improve the quality of information of the performance and genetic records. So that gives us like data-driven decision making that helps us to do the decision making using the real-time farm information. So before we just when we analyze the data, we did the milk and milk composition. We only consider these traits, but now we are considering more traits so that we can have more better evaluation of the animals, so that we can have shorter calving rate, high conception rate, age at first calving, shorter age at first calving, something like that. We are considering all those aspects. And together with that one, we are also doing some genomic selection. We already started doing genomic selection in the cattle, and we are planning to do this in other species as well. So we have trying to apply this genomic selection in cattle genetic improvement program to develop the reference population to estimate the relationship between genetic marker and also to the traits. So this is the very robust way of doing the genetic improvement in the livestock sector. So apart from that, we also focus on the crossbreeding, so that we introduce many different breeds. This is another example for the livestock sector. We introduced many different breeds of goats, and we did the crossbreeding to optimize the meat production to meet the national demand. So this is what we are doing, and we have done this in different agroecological zones to figure out which blood level of different breed is appropriate for where. So in case of Boer crosses, we found 50% Boer and local is appropriate for the meat hills under the traditional farming system. But if you have the semi-intensive farming, then 75% also is good. Yeah, when you say this is better, that means the growth rate is also better, and also the survival rate is also better in case of this group. So, we recommend 50% for meat hills traditional farming and 75% for semi-intensive farming. So, but when you see the indigenous curry goat, we found that only the growth rate is not that, in comparison to Boer goats, which is developed for meat breed, developed as a meat breed. When we compare with that, the curry has other aspects like reproductive performances and adaptation aspect are better than the Boer goats. So we also need to think about how to improve the curry goat as such, and also we have to take another strategy like for meat production. We need to do the crossbreeding not with the curry goat, with the nondescript type, which we don't know which cross is that. It is cross of different breeds. So to gain the faster genetic gain, we are working in the different technologies, assisted reproductive technologies in goats, and also we are also working in cattle and buffaloes and other aspects. In goats, we use this technology and we achieve up to 55% success rate in the conception, but it can be improved by improving the management as well. And we are also working in the fish, in the pigs, using the fresh semen, and which is very encouraging, which gives a very encouraging result. About 84% have the success rate. But when you see the other literature review, it is even higher in other countries. So we also need to work on this to improve that one. So for sustainable agri-food system, climate change is really, really important. And also, so we need to work on the climate smart livestock genetics. So in this case, we need to we need to do the breeding, and we need to breed animals for heat tolerance, disease tolerance, and parasite resistance. And in this case, Nepal Agriculture Research Council, we are working to develop the breed in cattle. Yeah, we use indigenous Terai cattle and cross with the commercial cattle population, cattle breeds, and we develop the crossbreeds, and we are trying to make stabilize that gene in that population so that we can develop a specific breed for the Terai region, which can tolerate the heat and also which can produce better. So that is what we are planning to do for that one, and the outcome of this one is to get the greater resilience to climate change and also stable productivity under different stressful conditions. So I just wanted to give one example here. I really like this slide because it shows that how it is so important to have the adaptive gene in the animal itself, because this is when we compare the growth of Boer, crossbreds of Boer and local and the curry goats, and in this one you can see this side pointer kuno. This side shows that in the earthquake time, when they have the adverse condition, that time the pure Boer and the 50% Boer shows the lesser performance when compared to the normal condition in the other side. So this, and when you see the curry goat and the 75% curry, then in that condition, the growth rate is not that much different. So this shows that the adaptive gene is so much important for the production, sustainable production of the animal. And that is why the indigenous animal genetic resources is so much important for the sustainable agri-food system. So we need to do the conservation of all the indigenous one for the support for including the gene of adopting in this climate, in this region. So there are so many breeds that we have 26 different breeds that we have identified and we have characterized in different level, like in production level and in molecular level and in biochemical level. In different level, we have characterized those breeds, and we are trying to see the what are the adaptive genes they have that we can use for further genetic improvement. So we have done some research in the high altitude to see to understand the mechanism, the molecular mechanism that that governs the high altitude adaptation traits in the livestock. And we did that in the sheep breeds of Nepal, and we compared that with all the sheep that is found in all over the world. And in this project, we found a novel molecular mechanism for high altitude adaptation in sheep at the Himalayas. And actually, we found this gene, which is very important for the adaptation in the high altitude. And when we compare that with the sheep in the low hill and the plains, they don't have this gene. This is very specific, and which is we call it like putative candidate gene for that. And we also did the gene expression. We also, after identifying this gene, which is which is related to various biochemical processes which have possible biological functions such as morphogenesis, along morphogenesis, angiogenesis, which is related, have crucial indicators for adaptive evolution. So this gene, we also see the gene expression of this gene in the adult animals, and we found this gene in high level. So we also work in the Nepalese buffalo and see the adaptive gene in the different breeds. And we found in higher altitude, in the hill buffaloes, and when we compare with the buffaloes in the plains like Muras and Tarai buffalo in Nepal and other buffaloes which is found, in the world, we compare all the data that we collected from the NCBI. We collected all and we analyzed, and we found that the animals that is found in hill regions of Nepal have very specific adaptive gene that is, we need to preserve, and we need to, when we do the genetic improvement program, we need to consider those as well. So we have not only did the genetic diversity work only, we also use the genes that is also have the influence in the association with the production. So we also work with the different microsat markers, which we try to see whether it is associated with the milk production in the buffaloes. We found out of 8 markers, 6 were associated with the milk production. So we can use that for the selection of the animals in the early ages. So we also work in different aspects, like to see the quality of milk in the indigenous cows, and we found that the CD cattle and the Lulu cattle, but we did the work in the CD and Lulu only, so we found A2 beta-casein protein in milk of these breeds. So we need to work on other breeds and other species as well. So we have to work on that, and we also work in the utilization of the indigenous animals, and to we worked to develop the dual-purpose poultry, and we use Sakini chicken as a base population. So this helps in leveraging indigenous breeds for sustainable livestock production. So we use Sakini and we cross with all the breeds that is, all the commercial breeds that is found in Nepal, and we see their performances, and we have developed different combinations, and we found 3 different populations which is better in case of meat purpose, in case of egg laying, and in case of fertility and hatchability, and also for the adaptation. So we found when we did the simple selection, we found that we have developed the 3 different lines that is good for different purposes. So even when we have just see the Sakini population, we found the variation in Sakini population, and with the simple selection, we found that the Sakini chicken is also not that significantly different to the commercial breeds in terms of eggs and growth production. We also work in the, not only in the this thing, we also work in the yaks and changras that is found in the high altitude, and for the first time we have done this very systematic analysis, systematic evaluation of the changra fibers, and we found that Nepal's changra fibers is as good as that is in China, Mongolia, and Afghanistan. They are called the finest Pashmina wool, Kashmir wool in the world. So Nepal has the good quality Pashmina, but we also try to see how to improve the quality and quantity of Pashmina as well as the meat production. So we did that by keeping them in the stall feeding during the time of lean period. In the lean period, the production is always, they go very low. The weight of the animals is so low that they just survive. So if you give the little supplement of feed, then the growth rate increases. It doesn't go down. So that not only improves their growth performances, that also improves the reproduction and other aspects as well. So this is what going on, and also we are working on the fake bee gene, that is fertility gene in sheep, small ruminants as such, and we found in curry goats, even though it's a prolific breed, we don't find this specific breed. So we need to work on this one to improve to see the mechanism for the prolificacy in the curry goats. But in Kage sheep, we found that there's a positive relation has been observed with the kids per doe, which is associated with the gene that is. So we can use that for the marker-assisted selection. So, our team, NARC and the AFU team are planning to work on this aspect in the small ruminants, and we already got the funding for this one to work further comprehensive study. So we are working in different genetic improvement so that we can develop the breeds which is cultural importance, and we are working using the indigenous breed with the exotic to find the black collar pig, which is have cultural importance in the eastern part of the country, community in the eastern part, and we also working on not only on the this genetic improvement stuff. In order to improve, I already mentioned that in order to improve the production and productivity, we also have to work in the feed research. We also have to work in the disease aspect. So we work mainly on the infertility, because if the animals are infertile, then we cannot have the result as we are expecting. So we are working on zoonotic diseases. We are working in brucellosis and leptospirosis incidents in the cattle and also in other species. And we are also working on the emerging and re-emerging diseases in pigs. And these are all the examples that what we are doing so far. And we are also working in the antimicrobial resistance, AMR, and we get the very dangerous situation, and that all the antibiotics that which are found are all used up and also getting the resistance along with that one. So when you see the challenges of this sustainable agri-food system, we have the challenges mainly is the climate change. Our indigenous genetic resources, we are losing them, and that is also the challenge that we cannot use the very important gene that they are having with them. So that is another challenge for us. The limited access to the advanced technologies, and we have started this performance recording, systematic performance recording, but this is not the comprehensive and continuous one. That is why it needs to be very continuous, and that is another challenge. And most of the farmers are smallholder farmers, so they don't have this quality breeding stock. That is another challenge, and also they are not eager to involve in the this genetic evaluation system. So that is another challenge for us. So there is many, and the major challenge is like we have very weak collaboration among the research, extension, and academia, and also the industry that is involved in livestock sector. So these are all the but there's still the other future perspectives, like we are now planning to do the genomic selection. We are thinking to work in the livestock farming that is for data-driven breeding decision, and we are also having the manpower, which is which they know about the new technology about like IVF, ET, this sort of thing, and also there is different international collaborations, and there are also different supportive policies and investments are there. But for all this thing, I think we need to come together. The academia, the extension, and the research. We have to come together. And all the research that I have shown is one way or another. We are involved together. Actually, the students from the universities, they are involved in the project. That is why we are successful to have this kind of findings that has the very practical implications. So that is why NARC also have the strategy, taken the strategy that for every project they give NARC gives the priority if there is a collaborative partner. So NARC has taken this strategy from this year that if we have some collaborative partners from extension or local government or from academia, that project is funded and given priority. So to collaborate with national and international research institution is our priority, and extension services and with education institution to develop the comprehensive solution of all the researchable issues that we have. So, and especially for genetic improvement, only one institution cannot work to get the concrete conclusion, right? So that is why this is again very important for genetic improvements aspect. So for future, collaboration offers wider opportunity to improve livestock productivity, sustainability, and resilience through shared expertise, technology transfer, and coordinated research efforts. So that is what we need to do. So when we think about having the better sustainable agri-food system especially in the livestock sector, a key outcome of the research is the strong collaboration and the coordination among research, extension, and education. So I urge you, everyone here present here, to come together, work together, and for the betterment of the farmers and livelihood. Thank you so much.
Thank you, Dr. Nina Mate Gopaliji. त्यसै गरी अब म नेक्स्ट प्रेजेन्टरलाई किनोट प्रेजेन्टरलाई यहाँ आमन्त्रण गर्दछु Dr. Kumar Lama from Kathmandu University. He is the head of the Department of Agriculture, School of Science, and he is the assistant professor and the head of the Department of Agriculture at Kathmandu University with expertise in horticulture research, teaching, and sustainable agriculture.
Hello? Okay. Up and down, होइन यो मात्रै Good morning. Good morning, all. Can you hear me properly? Okay, I am Kumar Lama from Kathmandu University. It's nice to hear all the presentations, and I like to speak less, so I would like to finish my presentation as in given time, try to finish in that time frame. Okay. So, before entering to my title, I would like to show this photo. This is the photo from 2018, just 8 years back from today, in the June month, in summer. It's like the weather of Chiton in Israel. And I'm picking apple there, like Sir Isaac Newton. He developed the theory of gravity after falling the apple. So at that time, my mind was clicked. And I remember my undergrad days. In 2018, I think it was the summertime again, and we watched the television at that time in the mess, and we heard a speech of one of our agriculture minister. I forgot his name, but he said during his speech, he said that we need to grow apple in Tarai region, and we all were laughed in the hall because we said that this minister even don't know where we grow the apple. Even he doesn't know we grow it in temperate regions. And in 2018, myself, I'm picking apple in hot regions, like the weather in Siton. So that's the clicking point. So that point gives me the feelings or sense of we need to transform or we need to change our thought and change our education process. I think our thought process can only able to change by the education, and we need to transform that education. So today, I'm talking about the transformation of Nepalese agriculture education for commercialization. As I said, I'm from Kathmandu University. So if you look about the education in Nepal, basically, we provided two types of education in agriculture. One for short-term course, or a specialized course for the agriculture. That's basically focused for the trainings. And the second part, we go for the longer course, which like
like here, which is provided by the university. So in Nepal, if you see currently, 8 universities are involved in agricultural educations, and they provided the education through the 35 different colleges. And if you see it distributed from the eastern part to the western part of regions, and based mostly, it lies in the Tarai regions and mid-hills of Nepal. And apart from this, if you go to the CTVT, as per the data of CTVT 2024, we can see 296 institutions are involved in short-term agriculture courses, including ISC agriculture in both stream plant science and animal science. And through that, 11,840 skilled individuals are produced per annum. So if you see these two numbers, it's a huge number. Either you talk about the short course or about the long course. So if you go to the timeline of agriculture education in Nepal, in 1972, ISTU was established, and it comes to IS Rampur in 1974, and it started the undergraduate education. And then it started the journey with extension of its different colleges, and if you see in 2010, something remarkable changes happened in agriculture education in Nepal. So if you focus in 2010, for the first time in country, the Nepal government has established Agriculture and Forest University through the Parliamentary Act. And that's the turning point, I can say. If you see this from the 2010 to 2021, the huge changes happened in agriculture education. And out of 35 agriculture colleges all over the country, 27 were established in this time frame. So that's the turning point. I think that's the boom time for agriculture education in the number of college, and could be in the number of students too. So if you see, though there are 8 different universities, if you see that their curriculum, we cannot see that much significant differences in curriculum in undergraduate courses. Everything, some like us, our PU, KU, or RJU, Rajasi Janak University, which recently started their agriculture. We try to be something different, but still we are same. There is not much differences. We cover with some basic courses, and if you see the credit loads, it started from 156 to 170 credits, depending upon the different programs, different universities. So it's a huge load if you see for the undergrad course of other streams. Even if you talk about the environment science, if you talk about the biotechnology, or if you talk about the engineering courses. So, and if you see the load in weightage of practical in this part, it goes from maximum 40%, and it starts from 19 to 40%. So we can see some differences in the preference of theory and practical classes in the curriculum. And still, though the number is less compared to the 170, 156 is less, but still this is a very huge load to our graduate programs. So after this, if you see, though the numbers are increasing, the number of colleges, and loads is very high, if you see the applications of Bachelor in Agriculture program, from the last 5 years, from 2018 to 2013, it's declining. And remember that, historically, agriculture education in Nepal is not the first choice of students. We were always, even when I was in the university at that time, especially the students from the medical science who were unable to succeed in that field, they come to for the agriculture, or from the engineering stream. So, historically, we are either secondary or tertiary in this part, in the choice of students. And at the same time, we are booming our colleges, and that reflection we can see by the declining number of applicants. And I can take the 2 giants name here, Agriculture and Forest University and ISTU. If you see the pollen applicants in AFU, we can see 62% in the 5 years, and 72.9% of applicants fallen in ISTU. So if you see the time of our applications applicants, is only 1/3 of students are interested only for application. That's directly impact for the enrollment, and not only enrollment, it also impact on the dropout rate. So for this, that's the application rate. And another part, if we see, our graduates, they are actually, we say it brain drain. They are trying to move as much as possible outside the country. They want to migrate. We took these 9 different reasons from the paper of these 2 gentlemen, Kartela and Sakota, from 2018. And then we also did the survey with the graduates or alumni of agriculture education of Nepal who are now in abroad, and we asked them reasons. And if you see, these are the 3 reasons they gave, the major reasons they want to migrate outside, or the reasons they were migrated outside. So the first one is academic excellence. I think could be reasons. We grow in the numbers, maybe we forget to grow in the quality. Another part is provision of scholarship. If you see after the master's, bachelor's, students wants to have scholarship so that they don't need to depend on with their parents for the tuition fee and their living cost. And another reason is their security. That's in the security, we especially we categorize it work and stay, job security, and some of them really wants to migrate abroad, so they want to have permanent resident cards in a few years. So these are the reasons our graduates are migrating, and at the same time, that the application rate is decreasing. So if you see the gaps, where is the gaps? So World Bank said in 2003, where is the gap? We see especially I take these 3 points in the gaps for the accreditation and quality assurance. Basically, we have the poor infrastructure. Usually, that's what people say, like classroom, laboratories, and field equipments, and also the recruitment and staff development. And the interesting part is pointed by Agri-Food 2020, though we are called new universities, from I can say, including AFU and myself, ourselves from the Kathmandu University, we started our journey from, AFU started a journey from 2010, and we started a journey from 2020. But although we label as a new university, if you see, our curriculum and teaching approach are not new. It's similar to IST. So we are not able to coming out till now from that mindset of 1972 at the beginning of ISTU. So for that, and also I think we have heard that in several programs, the dedicated council, we demand for the dedicated council. So far, even we have almost 22,200 seats for the undergraduate program. Still, we don't have any dedicated councils to monitor it. So that's the gap in the quality assurance I like to highlight here. And what is the reforms we need? Is that actually necessary? Yes. If you see, if we go to the 2 decades back from now, even the World Bank in 2003, they suggested to go for interdisciplinary production marketing focused curricula rather than focusing on conventional curriculum. And if we take the examples from our neighboring country, from India, ICAR 5th Dean's Committee report and National Education Policy 2020, that's ensure the flexible, robust, and globally relevant education. And that relevant means, what does that mean? That means you have to focus on these 2 parts, employability and entrepreneurship. And I think most of us know that ICAR set the standards of agriculture education, forestry education, and biotechnology standards in India. So we don't have that type of mechanism in Nepal. At least we have to maintain this standard. So that's the difficulty, reforms we need to know. And another part is always after the graduation, students want to have job. They want to have a secure positions. So if you see in the previous data, people like to go for government job before, because there is the number of vacancies are higher, but the stage is declining. But our approach, our curricula is focused for, public service commissions job more than compared to the private sector jobs or entrepreneurship development. And it's pity that when I meet the people from the private sectors or from the agriculture companies, they always complain with us one thing that, okay, it's a graduate from the university, but they don't have even the basic skills these days. So I think we have to focus our students or our graduates, develop our curriculum for that, so that they can also go to the private sector or they can create the entrepreneurship by themselves. So for that, as a university, I did the review in a place, universities, business incubation or startup mechanism, and recently, from I think 4-5 years, from the last 4-5 years, most of the so-called leading university, I can say, we established business incubation center in our university. And also we provided some amount of startup fund to the students or competitive student ideas to materialize their ideas as a business. So that's the way we have to go, and this is also directed by National Agriculture Policy 2004 and National Agriculture Policy 2019 of Nepal. So here, what I suggest, university need to have agribusiness centers. The centers should be focused on counseling, startup grant, market research, need assessment, and training, and other things. We need to support our graduate students to establish their entrepreneurship, and then we can also coordinate for that government and different research institutions, including NARC. And if you see the technology we can work on from the different parts of agriculture, from crop production to the post-harvest to IPM and everything, so we can go there. And that technology should be transferred to the cooperatives or different private sectors or different government agencies, and that can develop as entrepreneurship, and that can help to the economic growth. So this is the model I suggest from the perspective of my team, but we are open to discuss on that. So after coming this, I would like to conclude that in one sentence, and I have a list of recommendations. So what I see with visiting the curriculum, we all are almost same. We try to be different. We pretend to be different, but we are all same. We are in the same boat. So we have yet to fully align their curricula with recent technology and advancement and emerging technology. That's the remaining part. So that might make who can adopt this technology in curricula first might make us different actually in the real field. So I have several recommendations. I put it in two different parts. The first part is, in the first part, agriculture curriculum, resources and entrepreneurship. And the first point we need to see here, current existing BSc agriculture program has even minimum credit hours of 156 and a maximum of 170. That's more than 5 years course load of other program. And we are trying to finish it in 4 years. So we have to think. And this generation, we call it Gen Z, I think they are not able to bear this pressure like us. So we have to separate it. Like as in ISC AG, if you see the ISC AG, there is a ISC plant science and animal science. And in BSc AG, we mix everything together. So I suggest to separate it as a BSc agriculture plant science and BSc agriculture animal science or fisheries, or you can add others program. And the credit hours should be limited in 120 to 128 credit hours for 4 years. This is the international standard, and this is practicing now. And in Kathmandu University, most of the program, they practices this type of curriculum structure, but still we are facing difficulties to go in that. And another part is, our BSc agriculture program that focus for the science stream students. And these days, people from the other stream, they want to learn agriculture. They want to come have a graduation in agriculture, and we don't give a space for them. There is no such program at develop to include this type of students. For that, we can go for some options like BTech in agriculture. We can include different program, bachelor in agriculture, and if needed, we can also go for professional programs so that these peoples can include. And if you see, actually, these peoples are actually working in this production sector of agriculture rather than we agriculture graduate. So this is the bottleneck, I can say here proudly, loudly. So, and in the curriculum, we must integrate the modern courses like agriculture, AI applications, related courses, and molecular biology. You know, when I hear about some people say talk about the genes, we say, oh, this is the work of biotechnology. No, that's the wrong concept. This is the basic thing. All the agriculture students should know these things. At least you should have basic knowledge of these things. So that's the concept we have to come out. And we also go for the business incubation centers, which I highlighted before. And each business incubation center should have seed money fund to support the competitive ideas of students so that from there we can also bring the ideas or materialize the ideas to the entrepreneurship. So I think this thing can help to think out from the public service commission job to the private sector employment or to establish the being an entrepreneur. And in the second part, in the second part, I like to talk about the recommendation about the national policy and cross-cutting issues. We need Agriculture Council, as I said before. We can give any name, whatever. We need a dedicated council. I proposed Agriculture Council, but you can give any name. And that body should be autonomous and has rights to monitoring physical and human resources of agriculture college, licensing of graduate, and suggesting periodic updating of the curriculum, as did by the ICAR India. And we must have, I think this is what I need. We need to have separate agriculture education policy, but that policy should come under the framework of National Agriculture Education Policy of 2019. Why we need this? Because from the day one, we have been taught that teachings goes to the university, research goes to the NARC, and extension goes to the ministry or department. So I put here very soft word. We need this policy for the breathing, but if you allow me, I can, or a little bit harder, that's we need to blend it. There shouldn't be gap in the understanding of these three sectors. And another part, we have to go for academic credit banking systems. So students should not need not to finish in 4 years. They can earn some credits and they go for entrepreneurship or for the job market, and then they come and they earn another credit. And within the given time frame, could be 7 years or 8 years, they can fulfill all the requirements by storing their credits in the credit banking system, and they can graduate. We have to move in that direction, which is also directed by the National Education Policy 2019. And through this, we can also, bring the farmers or agro-entrepreneurs through these certification systems by this credit banking system. And last but not the least, we have to also focus on cross-cutting issues like disadvantaged groups, women, conservation of neglected crops, livestock, and indigenous knowledge should be addressed in our new agriculture courses curriculum. So in this way, I would like to wrap up by thanking my colleague, Janardan Dikari, Ajay Jaishi, and Roshan Subedi, who helped me to save this presentation up to here. And it's nice to be here in AFU. Thank you for inviting me as a keynote speaker. Thank you.
Thank you, Dr. Kumar Lama.
ल ए rhythm in yak भन्ने छ है अच्छा Okay. Yes, thank you. So, good morning, dears. I have a very good privilege, and I thank the organizing team to provide me this space for this keynote speech today on 27th June, entitled Biological Rhythms in Yak and Their Role in Nutrition, Reproduction, and Production. So, let me first introduce this yak. He is a mountain dweller, and is the best flagship among the mountain animals in our country. So, we have yaks in Central Asia and China, South Asia, some South Asian countries, including Nepal, India, Pakistan, Bhutan, and Afghanistan too. So, so this is my own figure that I dwell the Annapurna circuit in Manang in 2008. So it is a very old, but I am going to show here is, sorry. So you know the place, where is it? If I've been in somewhere in Manang, Mustang, Gorkha. So if you know this mountain, then you know the space and available for grazing yaks. It is somewhere in Manang. I don't know the exact village name. So this is the resource where these kind of animals are grazing and surviving their life and helping to enhance the livelihood of the mountain herders. So let us go to the evolution of yak. It is one of the youngest animals on the earth evolved, but it started from the same genus, both Primogenes, not less than 2 million years ago. And the history of its divergence starts not less than 2.2 million years ago. That is very close to the tectonic rise of the Himalaya. So there are some species left at the top, and they become they are regarded as yak genotypes in the new science nowadays. So it is one of, we don't have nomadic pastoralism yet, because these animals are still rare in so-called transhuman system, and this transhuman system is started due to the type of the tectonic rise, or when Indian plate moved to the Eurasian plates, then there are some surfaces raised off, including Tibet and from the Shivalik zone to Tibet. But these animals can be confined to these high altitude regions of the world, and so, and these animals are basically confined to the very cold regions, very harsh regions, and they can survive maximum, tolerate up to maximum 15 degrees centigrade to minimum minus 45 degrees centigrade. It is too harsh, and they can adjust themselves to the limited oxygen availability. And just remember that about 1/3 of oxygen availability is declined at 3,000 meters. And if the animal surpasses near 5,000 meters, then we can expect how much the hardship of oxygen is there. And they are adapted to seasonal vegetation patterns. If the altitude range is too diverse, then we can expect there are diverse vegetations and withering and other patterns of the vegetation. So let us come to see the circadian rhythm. That's today's talk, and it is simply a biological physiological behavior process, and that is indigenous biological timing system and for the mammals, and it is related by regulated by the suprachiasmatic nucleus, SCN protein, and it is synchronized by other environmental cues. And this circadian rhythm is critical for homeostasis in many domestic animals.
So I don't go much, then the importance is the physiological adjustment of yak. That's close to that exhibits unique adaptation to hypoxia, extreme cold, and seasonal photoperiod variation. So I mentioned my scope of presentation today, including different old and new publications, not less than 35, and we make different criteria of inclusions. And let us say this yak has the circadian rhythm regulates feeding behavior that controls hormonal secretion and affects productivity and reproduction, that impacts animal health, and it is important in management, deriving the health management strategies. And let us see one example, that there are two harsh environments. There is no feed, and there is feed but under the ground with totally withered. So these are the common grazing strategies of yaks. They can graze, I don't say simply grazing, but they can scavenge even in very harsh conditions. And behind this, there are some physiological basics of adaptation. One is the high hemoglobin affinity to high altitude, efficient oxygen utilizers. They have the low metabolic rate. That means in winter, their body size declines. And in my own record, yak reduces its body weight up to 20% in winter. That is one kind of adjustment. And behind it, there are some other physical, biochemical, and anatomical structures different than that of the normal domestic, we used to say like cattle, sheep, and goats. And they have certain different energy conservation strategies that can help yaks to adapt to very high altitude. So then we can make the yak study model, and for this we can say we can report to the unique chronobiological responses to record and different body vitals than cattle and sheep that we can measure in the upcoming research. So it is already adapted. Okay, then let's go to the environmental modulators of rhythmicity in yak. The first thing we talk is this rhythmicity is sometimes maintained by the feed surplus, okay? And rhythmicity happens on seasonal, that is regulated by seasonal change and the daylight available. In winter, there is low metabolic activity, but in summer, there is high metabolic activity, and this metabolic activity slowly declines towards the onset of winter, and in autumn, it is nearby peak, but after summer, here we say it is nearby autumn, but not actually on summer. So there are three modulators. One is the photoperiod, second is the melatonin secretion, and third is the nutritional adaptation itself. So here are some examples of the related studies. One says circadian rhythm of melatonin. There are many, and other is that is melatonin release and receptors, molecular characterizations, a tissue-specific distribution of melatonin. There are many experiments that has been already done with respect to the circadian rhythms in yak. And most interesting one is to the body vitals, which body vitals work on the what, and also it has some relations to the fertility of yaks at the last. So here is one molecular clock genes that regulates the mammalian circadian clock. It says during the daylight, when the daylight is offered, there is transcription of some clock genes that activates the DNA, PER and CRY proteins in DNA, and through translations that accumulates more protein during the day. And also in night, this is reduced, and there are some feedback loops even in the daytime. So this is the greatest experimental part that is developed in light of yak research. So this is the one important part is this protein ratio, the clock to BAMAL1 activity that makes a complex feedback and that recites the circadian rhythm in yaks. Let's go to the non and photoperiod sensing pathway. If we talk, then photoperiod light is important part that activates the retinal protein at cells. Then it activates SCN, and finally it activates the pineal gland. And pineal gland would release the melatonin at night, mostly at night. And this production of melatonin is always higher at longer durations and night, and shorter nights that has very low melatonin production, or the melatonin production is very low. So this is one of the example that sets the some circadian rhythm and the gene expressions. And there are many experiments I have summarized here. And AXO altered gene expression, then PER2 variation observed is the main reason for this circadian cycle. And adaptation to altitude influence of hypoxia, seasonal gene expression changes, they are all related to melatonin functions. So, here are examples of the physiological effects of this rhythmicity. One is the behavioral rhythm, second one is the metabolism and thermal rhythms, other is the regulation of endocrinal rhythms, and finally we can say the reproductive rhythms. Those are related to this rhythmicity. So in integration, there are some good examples of the clock genes and the photoperiod response. And there are these all leads finally to the seasonal traits of yak. Then here are basic features of yak adaptation to altitude and cold stress. This rhythmicity starts with the environmental stresses, then it affects the feed intake and grazing behavior. It is seasonal, and there are some differences in rumen fermentation patterns, and this fermentation pattern is led by different kinds of microbes, and that led to differentiation of energy cycles, energy metabolizers, the environmental stressors like methane, ammonia, and other changes. So they are all related to feed and season. The hypoxia later affects physiology, low temperature stress, photoperiod variations, limited nutritions, and seasonal changes. They are all affected by the rhythmicity. So, in common, we can say the clock genes triggers to signal to metabolic genes, and later that triggers fat storage in winter and its mobilization, and later that helps to the winter survival. Then let's go to see the dietary shifts in yak leading to variations in rumen. If there is dietary shift or the shift from one season to other, or from lean season to winter dry season, this leads to variations in rumen microbes, and that has some shifts in microbial populations or microbial quantity. And this leads to some functional adaptations, example that helps these enteritis or these bacteria groups help to high fiber digestion, high energy efficiency, metabolism in summer, and more enzyme activity during the release of, due to the release of cellulase and gyalanase in digestion process. So later on fermentation in the rumen, these metabolites, these metabolites are very important, and the host adaptation is also important. Yak or any kind of ruminants, they don't produce their own energy themselves, but they are sourced from the bacterial fermentations. So the bacteria are hosted by the animal, and the bacteria in return they supply the energy. So about 60% of total energy is the bacterial energy, and about 40% of, only 40% of the energy that comes from the substrate fermentation in the rumen. So then it overall leads to the yak performance, let us say to the weight maintenance in winter, the milk production, and survival and fitness of the animals. And there are other impacts, example, impact on ox overall performance, energy optimization, the rhythmic intake patterns, and they are further influenced by the ambient temperature. So here is one interesting part, and the in our case, if yak is the first leading purebred and urang is other hybrid with cattle and dimzo is another hybrid cattle and cow is also purebred. So if you see the literatures, the nitrogen use efficiency is always high in yaks, followed by dimzo chauris, then later by urangs and the least in cows. So this is nitrogen use efficiency patterns. And amino acid conversion and production of isoacids are similar pathways in BFE production from carbohydrates, but nitrogen use efficiency is different. That's the mere typical part of the metabolic differences in yak than in other domestic ruminants. So this is normal summary. We can say in the protein breakdown, there is release of the microbial breakdown that releases microbial protein, and finally microbial amino acids. They could be utilized by animal, and some would be lost through ammonia. Yak is not the animal that holds all protein. There are also some losses, but these losses are reported, the ammonia and methane, these losses are reported less than any other domestic ruminants, and they are lower than the IPCC default values. This is the importance of rearing yak in the Himalayas. So, then let's go to talk about the hormonal rhythms. Melatonin we talk, cortisols, thyroxines, T3 thyroxine and insulin. They are all different at different time and functions. Okay, and they have certain environmental imprints, and they are recorded by many research. They are already established. So then we already talked these longer nights have higher melatonin, certain nights they have lower melatonin duration. I mean this melatonin production is low. So they are all effects all together, and at high altitudes this system is finely tuned to cope with extreme light, dark cycles, and modulations may involve altered amplitude of phase of PER-CRY genes and ensuring optimal metabolic and thermoregulatory responses under harsh environmental conditions. So let's go to the behavior rhythms, and there are some differences in grazing to scavenging that I have already shown to you, and that leads to the environmental adaptation. Next one is the endocrine rhythms. The melatonin peak I already showed you, cortisol peak, thyroid peak, the photoperiod influences on hormones, and that overall regulates the metabolism. And in terms of melatonin rhythm, there is secreted during darkness. I already expressed it. Controls circadian timing, regulates reproduction, and that is further the indicator of biological clock. So then go to the hormone cortisol rhythm. It is peak in the early morning. This is not at night, and this is stress-related hormone that shows the daily fluctuations observed, or it is dependent on the daily grazing activity, or how long it goes for grazing, or how far it goes for grazing. Then it is influenced by altitude and regulates metabolism, energy metabolism. So in overall energy metabolic pathway, you see the energy use varies daily based on activity, and adaptation to cold stress, efficient nutrient utilization synchronizes with feeding, and that supports overall survival of the yaks. Then in terms of thermoregulation, it is simply the maintenance of body temperature, and yak is adapted to cold climate, and season variation observed in many studies and linked to metabolic rate, and it is all of the important rhythm for the survival of the yaks. Then this is one of the important part. We have some breeders here. It is very useful to you. If I work with you or if you work with me, that might be the important part for the research, the reproductive rhythm. Yak is naturally the seasonal breeder, and it is short day breeder, photoperiod dependent. It has hormonal regulation of breeding cycles, and reproductive efficiency is based on all clocks and all metabolic pathways. So here is one example of research that shows the season of the year influences semen output and testosterone in yaks. Because I am showing this, because these hormonal influences may be typically linked to the so-called heat stress in high altitude. Then it says the plasma testosterone varied by season, and this plasma testosterone is always high in the winter. When the yak becomes robust, then it may have high testosterone, it shows. And in other research, it says semen quality varied by season, and it is always good during autumn. When the yak is robust, the semen quality is good, or it is well linked to the nutrient metabolic cycles. Then other is testosterone and semen quality that shows seasonal patterns, and it is always, the semen concentration is always good near autumn, and the testosterone volume is always good at the end of the autumn season. So we can say the highly sensitive, it is one of the highly sensitive research, and we can say seasonal change in semen quality would be associated with changes in plasma testosterone, and this hormone regulation would be further regulated by the seasonal clocks and the feed available, or the feed we use to feed them. So then let's go to the photoperiod effects. There are some major environmental cues. There are many other environmental stressors that controls melatonin secretion, that influences reproduction, affects activity pattern or daily grazing pattern, and in overall seasonal variation of outputs, different outputs. So let's not go this, we have repeated it. Then hypoxia effects. Then low oxygen environment is responsible for the overall performance of yaks, that affects gene expression. Coupling between melatonin and active timing, and they also enhance conservation in winter. So then let's go to the compare this circadian and behavioral rhythms in domestic animals and the circadian rhythm in domestic animals that is in common for most of the species. But yak has some special adaptation regarding to other domestic animals in terms of feeding rhythms, in terms of rumination timing, light and photoperiod sensitivity, the seasonal rhythms, activity patterns, and the endocrine responsiveness. Okay. So then we can interpret like this. Light is the major factor that influence retinal muscles. Then they release a protein that releases melatonin rhythm, and in overall finally they have effect on gene expression rhythms leading to the performance of reproduction, metabolism, and hair growth. So then basic application, this rhythm in study we can use in improving feeding strategies, better animal health monitoring, optimized reproduction, and climate adaptation strategies research in the future, and also the enhanced production in overall. So the gaps remain here up to circadian rhythm studies are, we have limited yak specific research. We have more focus to physiology rather than rhythm. Then we need, it needs for molecular and chronobiological insights, essentials to look over underlying mechanisms of their unique adaptations, and translational and applied potential of research may be to be focused on leveraging chronobiological insights to enhance productivity, health, welfare, and reproduction in yaks. In conclusion, we can say yaks show strong circadian rhythms and adapted to extreme environments. Photoperiod is the major regulator, and there are still limited research available and in terms of biological perspectives. So let us work together. There are many phases to be saved in the future, maybe this kind of phases, and in this kind of harsh environments to work. And we are open to work, or we are open for any kind of contact. So thank you very much for your attention.
So we are heading for the last presentation, keynote presentation, but before moving to presentation, हामीहरूको one of our co-organizer, अलि छिटै निस्किनु पर्ने भएको हुँदा उहाँलाई हामीले मागेको चिनो प्रदान गर्ने कार्यक्रम राखेका छौँ। चिनो प्रदान म डाइरेक्टर डाक्टर उज्जवल तिवारीलाई यहाँ आमन्त्रण गर्दछु र मागेको चिनो ग्रहण गरिदिनु हुन केयरको टिमबाट केयरको रिप्रेजेन्टेटिभलाई पनि म स्टेजमा आमन्त्रण गर्दछु। Thank you, Kher, for supporting. So, for the final presentation of today and the National Conference of Sustainable Agri-Food System, I would like to request Professor Dr. Sundar Tiwari, Dean of Faculty of Agriculture, Agriculture and Forestry University. He is the researcher in agroecological pest management with expertise in habitat management, biological control, pollination ecology, and sustainable IPM process. Thank you. Sound छैन भाइ?
Hello. Yeah. Okay, thank you very much, everybody, for such a wonderful presentations and your patience until the last presentations. So today I'm talking about the IPM, especially in you might heard IPM in agriculture, but I'm thinking why not the IPM in bee colony? So that topic is under my presentation. So in agriculture, actually, so how many of you heard IPM, integrated pest management in agriculture? Yes, that's nice. Then what about the IPM in bee colony? IPM in bee management? That's why I'm going to raise the voice, and I'm going to penetrate that idea into the IPM in bee colony as well. So you might know, even you are familiar with the use of the chemical pesticide in agriculture, especially. So we have been using that chemical since 1967, actually in Nepal in 1997. Then after, until 2026, the pesticide use has been increasing. So we use normally pesticide in agriculture. That's why the concept of IPM has been arise. So So that concept, why not to multiply in another areas, especially in the honeybee colony, because there is also the risks of the pesticide in honeybee colony as well. So what we thought, so if we adopt those IPM practices in honeybee colony, and of course we can reduce the pesticide consumption in the colony, and also we can produce a very good quality of honey, because this is actually the urgent demand of the honey, we call honey in the global market and even the local market as well. So the technique is IPM techniques of pest management in bee colony. That is the gateway of advancing honeybee health and honey quality. Next. Okay, so this is my kind of presentation outline. I will be discussing about the pesticide use in agriculture and IPM practices in agriculture to reduce the pesticide and honeybee IPM, and where is the risks of the pesticide in honeybee colony especially, and what are the strategies that we follow in the IPM practices in bee colony, and some of the practices that can help to reduce the pest in honeybee colony and what are the overall benefits. So that is the trend of the use of the pesticide of the globe in Nepal. Actually, we started to consume the pesticide in agriculture since 1997, and we have been using that pesticide every year, and that pesticide use has been increasing. So we have been using about 1,664 metric ton of pesticide. That is the actual active ingredient every year. So normally in agriculture, we normally use for the pest management, basically in the insect pest management as well as disease management and weed control. And that pest is not targeted to the pest. It's a very minute amount, very tiny amount of the pesticide that that target, and the rest of the pesticide normally goes to the environment. So then the concept of IPM came in 1967. There was a globally adopted definition of the IPM came in that was developed by the FAO, and it has been said that integrated pest management is an integrated approach. So where you utilize that utilize not only the dynamic of the pest, also utilize the environmental conditions, and that can help to reduce the pest population below the threshold level. And in Nepal, we adopted that IPM practice since 1997 and 1998. So if you see on the picture, on the IPM pyramid, sorry, any issue in the presentation slide? Yeah. Sorry, some technical issues. Yeah. So this is IPM pyramid. Basically, in IPM pyramid, we normally focus on the sorry. Previous slide, maybe. The one more. Yeah, that one. The green part, that is the base of the pyramid. Basically, that is focusing on the cultural. The second yellow part is focusing on the biotechnological approaches. And the chemical part is getting the less priority in the IPM. Okay, so this is the point that the honeybee IPM is about. Honeybee integrated pest management, that also integrate all possible methods, starting from the cultural whatever we have, and also focusing on the regular monitoring of the honeybee colony. That help to improve the bee colony as well as increase quality honey. As well as reduce the pesticide and decrease the pesticide resistance. Basically, if you see on the honeybee colony, there are some issues, basically some insect and disease problem. For example, varroa destructor, there is a varroa mite. Then we have another big problem of the tropiolapsis mite, and a small hive beetle is emerging issue nowadays in the honeybee colony. And we have also the problem of the wax moth. and some disease, some European fall brood, sac brood, chalk brood, stone brood. These are the some fungal as well as bacterial as well as viral disease. So because of that problem, normally farmers they use a chemical pesticide. So that chemical pesticide is going to be making some unhealthy honey, and which is not safe to the human beings. So normally for the control of the disease as well as some insect in honeybee hive, basically farmers they have been using the epistant. This is the synthetic pyrethroids group of pesticide, and some epistol. These are the some common pesticide is has been using in honey hive. So the alternative to reduce that chemical pesticide in honey hive is could be the IPM approach, because when the pesticide is already in the honeybee hive, then it can reduce the quality of the honey. Then then we can get the pesticide contaminated honey. Also effect on the food on the brood, because that chemical pesticide affect on the brood of the honeybee colony, as well as, and if effect on the brood colony, then of course it can reduce the honeybee colonies as well, the number of the colonies, number of the bees inside the hive. And if you frequently use that epistan or some other chemical pesticide, then of course it can increase the resistance to varroa. That means farmers or even the commercial farmers, they frequently use that chemicals. That means the dose of the chemical can increase over the time. So the strategy that we thought is a first identify the pest, what the pest is there and the honeybee hive. Then we have to think about the regular monitoring, so that we can get the exact situation of the field, what kind of pest is there, and what is the intensity of the effect of that pest inside the hive. So that we have to monitor, because that can help to guide the management strategy of the pest. And also we can also get some threshold level after the regular monitoring of the pest, because that threshold level helps to guide the when to start the pest management practices inside the hive. And we can get out of the several pest management approaches, and we select the best one, which one is very efficient and effective. So the selection of the control methods is also is very important. And finally, how does that control methods is effective? We can evaluate in terms of the cost, how much cost happen for the adopting that control. So that strategy should be followed for the integrated pest management of the honeybee hive. So the principle that I shared before, so in honeybee IPM there should be some preventive approaches, there should be some cultural approaches, some should be some mechanical, some biological, and finally should be some chemical approaches. And that chemical, but nowadays what is happening, chemical approaches coming in front. That makes some unhealthy honey production practices. So IPM is necessary. Basically, we want to reduce that chemical pesticide in the honey hive, and we also improve the honey quality, protect the colony health, because if we spray some chemical pesticide inside, then it ultimately affect the honeybee colony, and also reduce the economic losses, protect the environment, because that is another issue. Then support the pollination. If we increase the strength of the honeybee colony, then the number of the bee colony will be increased, and that can ultimately supporting to the pollination. So the first step of the IPM that we normally says is a regular monitoring of the bee. Basically, if we inspect the honeybee colony even in the 7 days or 14 days interval, we can exactly know what is the situation of the hive, and we can, on that basis, we can at least try to find out where is the problem, what is the problem, and what methods what methodological strategies should be adopted. So that kind of practice we have to follow in the step 1. The second one, there are many preventive strategies are there. So on the basis of the regular monitoring as well as the regular observation of the hive, we can observe what things is necessary for the honeybee pest management. For that, either it can be a problem of the some adequate nutrition as well as clean water. Rotate and replace the comb. We can also replace the comb as a situation. Provide good ventilation and sunlight. That can also affect the honeybee colony inside the hive. And we can also maintain the strong or healthy colony on the basis of the observation, and also what kind of disease or mite resistant bees are inside the hive. That can be follow in the prevention strategy. Then the cultural strategies after that, we ready to management, practice good apiary hygiene, because this is very important, because from the perspective of the safety issue, microbial safety or some of the handling issue would be there. And replace the old comb regularly, maintain proper hive spacing and ventilation, remove weak or heavily infected colonies, avoid practice that spread pest and disease. These are some cultural practices. There are some other cultural practices as well, but these are some practices we can adopt in the cultural pest management practices in the honeybee colony. Then mechanical, especially if you are talking about the mite issue inside the hive, and because the varroa mite is normally affect on the drone bee. If the affected bee, drone especially, if you case inside the drone trap, and that can help to reduce the risks of the mite problem inside the bee colony. So that's why from the beginning we have to monitor, and on that basis we have to decide what to be done in the pest management, especially in the honeybee colony. Then chemical, there are many chemical approaches. So there are already episton is very common practice of the mite management especially, but there are some other alternatives which are non-chemical methods. Formic acid is already affecting the bee colonies if the concentration is very high. Even the oxalic acid is very good for the mite management if we maintain the concentration as well as the time of the application. And thiamol, there is some essential oils. So there are some alternatives which are non-chemical methods. Even some essential oils that can help to reduce the mite issue inside the hive colony. So garlic oil also is going to be affected, and even oregano oil, neem oil, basil, geranium and uracil. These are some alternative approaches compared to the episton, because episton is already a synthetic pyrethroids group of pesticide, and it ultimately affect the bee colony. That's why there are some other alternatives. There are some essential oils which can help to kill some mite population if we maintain this concentration inside the hive. So this is the some IPM approaches when the bees are affected inside the colony with the chemical pesticide, but there is also some alternative approaches. If what happen if the bees is coming from outside with the pesticide, because bees normally take the nectar from the outside. That time, if the pesticide is already contaminated with the nectar, and that nectar that can bring to the hive. So in that case, how to protect the bees and how to make the honey actually free from the pesticide. So IPM practice also should be followed in the agriculture field, because there are so many of IPM approaches in the agriculture. So that is the only one strategy that can help to reduce the pesticide in farm. So then if there is somewhere pesticide is already in the farm, then we can at least make some distance from the pesticide use area. We can also move away that hive from that area. And if some pesticide is already in the flowering plants, the bees can be confined in certain locations so that the bees cannot escape from the hive. So these are some protective measures. Or if there is already IPM practices or organic practices, then we can protect the bees, and we can also get the very good nectar from outside. So these are some approaches that can make the honey that we are consuming, that we can make free from the any contaminants like chemical pesticide or some microbial contaminants. So summary, honeybee IPM studies is the approaches to manage the pest and pesticide in a sustainable way. There are many chemical approaches. That chemical approaches is not going to affect to maintain the good colony. Also, that chemical has the possibility of the contaminating with the honeybee. So, non-chemical studies should be adopted. And finally, honey IPM improves the honey bee health, strengthen the colony, reduce the pest infestation and improve the honey quality. So, this is my presentation, I guess. Thank you.
Thank you, Professor Dr. Sundar Tiwari, sir. This is how we conclude our keynote presentations of the day and the overall conference. हामीहरू अब हाम्रो प्यारलल सेसन्सहरू चलिरहेको छ, पोस्ट इभ्यालुएसनहरू पनि भइरहेको छ. सो हामीहरू यहाँबाट अहिले विल मुभ टु दि प्यारलल सेसन्स, जुन कुनै प्यारलल सेसन स्टार्ट पनि भइसकेको छ. सो आई रिक्वेस्ट एभ्री वान टु प्रेजेन्ट टु देयर रेस्पेक्टिभ एन्ड टु देयर इन्ट्रेस्टिङ प्रेजेन्टेसन टु द रेस्पेक्टिभ हल्स. त्यसपछि हामीहरू लन्च लिएर एक्ज्याक्टली 2 बजे we will come here for the closing ceremony of this 3 days national conference on sustainable agri-food system, SAFS 2026. Thank you.